Okay, everyone. It's my great pleasure to introduce Professor Yolande Pijnenburg. It's a real pleasure to have her here with us. She's a professor of young onset dementia and a cognitive and behavioral neurologist at Amsterdam University Medical Center in the Netherlands. She leads a specialized neuropsychiatry clinic dedicated to the assessment of behavioral change in older adults, helping to distinguish FTD from psychiatric disorders, and her research has been really informative in this area. It spans the clinical, genetic, pathological, and molecular mechanisms underlying frontotemporal dementia and other young onset syndromes. Professor Pijnenburg leads the Neuropsychiatric International Consortium for Frontotemporal Dementia and the Right Temporal Variant FTD Working Group. She is also a key investigator in major Dutch national programs developing diagnostic tools and advancing our understanding of young onset dementia. Today she will present her keynote address, Progress in Frontotemporal Dementia, Time to Think Differently. Please join me in welcoming Professor Yolande Pijnenburg.
Thank you so much, Emma, and I'm really very honored, and it's a great pleasure to be here. And I've been enjoying all your talks during the last few days. So here are my disclosures. And I just wanted to start, unfortunately without subtitling, I would like to give you a little insight on what a patient with frontotemporal dementia looks like.
Langs mij en die hond hier liep. En toen kwam die hond en een beetje met mijn been. Ik tikte hem, was je godverdomme klootzak, zeg ik tegen die kerel. Die moet je aan de lijn houden.
I just showed it very briefly without subtitles, but the person was telling that he was being bitten by a dog, and he said, "God damn it," and he was very lively in explaining the story. But he had normal language, and he was also very interactive in the dialogue. But if you have him a little bit longer in your consultancy room, he will keep on talking. This is the same person at home.
En dameschappen doen het permanent.
So actually it looks like we're going to have a party here, but this is dementia. It's another form, another expression of dementia. And if you have a closer look, I have to put it a little bit more back. You can see how I do it. You can see the jigsaw puzzle he's making every day, and you can see his brain. There's severe temporal atrophy more on the right side than on the left side. And this person has a different style of management when you want to bring him for daycare, for example, because he's doing the jigsaw puzzles all the day. So in this presentation, I would like to talk with you on the three questions that is also depicted in Gauguin's famous painting, D'où venons-nous, qui sommes-nous, où allons-nous? Which means, where are we coming from, who are we, and where are we going to? I want to translate this to FTD. A little bit of history, where are we coming from? So you might have known that frontotemporal dementia was first described by the Czech psychiatrist Arnold Pick, and he described a person who had actually progressive on aphasia, and they found on gross mascopography in the brain that there was lobar atrophy, which was never described before. And then Alzheimer, who gently gave the name to Arnold Pick, saw in the brain these agrarophilic inclusions. Now we know this is tau, and they were called Pick bodies. So for a certain amount of time, the disease was called Pick's disease. But in the late 80s, there were two independent brain banks, the Lund and the Manchester brain banks, who found that not all patients who had dementia had Alzheimer's disease. There was a group of patients that had Pick's disease, but there was also a group of patients that remained. They had dementia, but they didn't know what they had because there were no inclusions, no apparent conclusions. And this type of dementia was termed dementia lacking distinctive histopathological features. And the time that I was doing my education, this was one of the types of dementia. I was not aware what was going on those brains. And then shortly after, Ian Mackenzie, who is a neuropathologist, has done a lot of work in FTD, found that these dementia lacking distinctive histopathologic features subjects do have ubiquitinized proteins in their brain. So there is a kind of protein, but ubiquitin is everywhere and it's a-specific. And it's important it's not Alzheimer's disease. And then in 2006, it was announced on a conference, an FTD conference, the protein underlying this ubiquitinated inclusion was discovered, and it was TDP43, TAR DNA binding protein. It was discovered by the group of Virginia Lee, and this was really great news. And importantly, it was not only found in frontotemporal lobar degeneration, but also in ALS. And very recently, I participated in an online meeting in a symposium that was held in the United States. And here you see Virginia Lee, who was still very proudly, 20 years afterwards, describing how this discovery went and how excited they were when they found it. So we're 20 years later now, and the pathological follow-up here was that there's a classification of Pick's disease, which is FTLD tau, but also other forms of tau that can be found in the brain, like threads and other types of tangles, which we all call FTLD tau. Then we got FTLD TDP, and a very small rest group where you find proteins, other proteins that can be stained with ubiquitin. And then a little bit later, there was a publication saying, well, we can subclassify the TDP, frontotemporal dementias, and there are 4 subtypes. And a little later, there was another subtype joining the classification. And so now, this is the classification that we are using, and this is actually describing what you see as a pathologist under the microscope. Everything that you see here, every shape is staining with a phosphorylated TDP antibody, but it really depends on which layer of the cortex they are in, if they are intracellular or extracellular, and which shape they have, and also if the white matter is involved, yes or no. So we got all these subtypes. And then you would think, okay, but what's the sense? What does it make sense at all? Can we keep on subclassifying? I mean, there's maybe more to subclassify. Does it have any relevance? Well, actually it does, because for type A, we know that everyone who has a progranulin mutation, it's one of the genetic causes of frontotemporal dementia, has TDP type A pathology. But if you have a C9orf repeat expansion, then you will have TDP type A or TDP type B in your brain. And semantic dementia, which is most often non-genetic, but genetically sporadic, has most often TDP type C in their brains. And these are all forms of frontotemporal dementia, genetic and non-genetic, and we don't know why these people have such different pathological features, although it's the same protein. And it's becoming very complicated, but I think I'll guide you through this a little bit, because if you see the clinical spectrum of frontotemporal dementia, and I don't know if I have a pointer. Yeah, you can see that there is the behavior variant of FTD, which is actually clinically the most prevalent. And we have got 2 language variants, semantic dementia and progressive non-fluent aphasia, where you see the brunt of the pathology is located in the temporal lobes. But we've got also an association with motor neuron disease, or ALS, in about 10% of FTD cases. And then we've got related disorder, corticobasal degeneration, and PSP that have also tau pathology and are often seen as part of the spectrum of frontotemporal lobar degeneration. But most of the syndromes, the underlying pathology is unknown during life unless you have a genetic genotype. And so it's very difficult during life to know which person has which pathology. And that's completely different with Alzheimer's disease, because if you've got Alzheimer's disease, we can predict that you will have amyloid plaques and tau-positive tangles in your brain. There's a few exceptions. Semantic dementia, in many, many cases, has TDP type C. FTD-ALS, almost always TDP type C. PSP and CBD, almost always tauopathy. But for the behavior variant, which is clinically the most prevalent, in most cases, we cannot predict the underlying pathology during life. And that's so important because as we still cannot do this, we cannot stratify patients for disease-modifying drugs, for example. That's still a very large problem, 20 years after the discovery of TDP. So to diagnose FTD, do we need to take the approach of dementia, because the D stands for dementia or degeneration? And I remind you that the definition of dementia is that you have to have functional loss in 2 cognitive domains, be it memory or executive function or visuospatial function. Behavior is also a domain, which you can count as a domain. So at least 2 domains affected, leading to functional loss in your daily life. You cannot exert your profession, or you cannot live well at home independently. And actually, dementia is an end stage of neurodegenerative disease. It's not the beginning. And the pitfalls in the diagnosis of FTD are that it's not like any other dementia that we envisage. In the first place, you will never see a patient with FTD coming to your clinic and say, I think I have some behavioral change, because they don't have insight. If they present like that, you can reassure those people immediately. The age issue, I will tell you about it later. And the non-memory issue. The complaints that they have are so different, and that comes with measurement instruments. How can you measure what they are complaining about or what their partners are complaining about? It's so hard to describe by words, also for us clinicians, and there are a lot of mimics. We know that FTD is relatively rare. It comprises less than 50% of all the young onset dementias, and about 10% of all dementias are young onset. So it's relatively rare, but many people come to see the clinic and say, do I have FTD or does my partner have FTD? But some causes are much more prevalent than FTD. And the lack of biomarkers, I will talk about. So there's a relationship with age in many neurodegenerative diseases, for example, Alzheimer's disease and Parkinson's. They really increase exponentially with age. ALS doesn't. It has a peak at around 70s, the 7th decade. And interestingly, although we consider FTD young onset dementia, the peak of age is also in the 7th decade. And interestingly for FTD, it's also highly variable in disease duration. So if you have FTD with ALS, you might live only 2 years. But if you have a very slow progressive FTD, which can, for example, happen when you have the C9 mutation, you can live for over 20 years. Is this still a dementia, you would ask? If we look at our own cases of our own memory clinic, here reported by Jay Fieldhouse, we see that the age at diagnosis is also having a maximum at around 65, and so the complaints have been starting earlier, so you might still consider people who are in their 70s as young onset dementia patients, but still we see that we were even diagnosing people above the age of 75. And what we also saw was that the older they were, the more memory problems they had. This could be due to AD pathology, but it's not fully explaining the clinical picture. And in FTD, it's not memory. It's mostly socio-emotional changes. And the thing is that lay people who are not well educated do not come to see you as a doctor and say, well, me or my husband has social-emotional changes. Is this dementia? No, that's not happening at all. So what happens, we cannot identify social-emotional changes as social-emotional changes. People keep on complaining, and we don't know how to measure them. And they keep on complaining, and we don't know how to quantify them. And they keep on complaining. And we don't know if these social-emotional changes are caused by brain disease. So this is a different world than doing a memory test and see if a person can repeat 10 words. We did a focus study where we asked partners of FTD patients what they thought was the most impactful symptom during the disease trajectory. And they almost unanimously described the stage where the social-emotional changes were there, which is mostly the very first stage of the disease, was very, very impactful to them. And many of them said that if there would be a therapy, the therapy had to be working even before those social-emotional changes were there. Because if you are going to treat a husband who has undergone a complete personality change, who you actually don't really like so much anymore, but you have to take care of that person, would you want to prolong that disease stadium for a few years? That's something that is very difficult to talk about, but many partners are telling you the same story. So then, do we need maybe a different trial approach in FTD instead of copying it from everything that's happening in dementia, especially Alzheimer's disease? And the diagnosis in practice is mostly made after this window of opportunity where the social-emotional changes occur has already been gone. And then, how can we make an early diagnosis of FTD? What can we do about it? Actually, until we have proper words, can properly recognize the social-emotional dysfunction, we can only do this in genetic FTD, I'm afraid. And there are criteria, they are actually, again, copying the Alzheimer's system, where we have MCI, and here we have MCBMI, which is meaning mild cognitive, behavioral, and/or motor impairment. This makes sense for genetic FTD, because if you have a therapy and you want to start it at the moment that the person is beginning to have very subtle symptoms, and that's the moment that you want to start the therapy, that makes sense. But if you want to see in the population who is having MCBMI, then you would maybe include many false positives, because FTD is a rare disorder, and it could be everything like marital problems or midlife crisis or depression, because if somebody changing a little bit their habits, this could be literally MCBMI. So that's very non-specific for the non-genetic population. And how do we measure the severity of dementia and the degree to which they are affected? Well, we have the CDR, which is widely used in Alzheimer's dementia research. But you see, it's not so well applicable to FTD, because in FTD, memory and orientation are not the greatest problems. So Dave Knopman said, let's attach something to it. Let's make behavior and language also domains of the CDR. So we created the CDR FTLD. Actually, still, I think it's a little bit artificial, and because there are so many other boxes, it's still difficult to capture what is really going on in FTD, and it's not FTD specific. From the grounds of Australia, this publication has been made by Miyoshi and others, and she made a really well-thought questionnaire. It's involving both behavior and ADL functions, and it's measuring the severity of disease in FTD and related syndromes. This could be an alternative for measuring the disease severity in FTD, although it's been used in some centers, but not widely as the FTLD CDR. Illustrating the symptoms of FTD is, I think, always nice to show with an illustration, because this is a drawing made by a patient with semantic dementia. And that's also the difficulty. It's not generalized dementia, but very focal. These guys can drive to the southern of France in their car without any problems. But they do not know what a strawberry is because they have semantic dementia. So it's very unequally distributed throughout the brain. They can make this very, very beautiful picture, and they're just copying it, but they don't know what they are drawing actually, even if it's the Taj Mahal. And if you ask these patients to draw a cat, a bird, and a fish, you see that the concepts that they have in their minds are already being vague. They cannot identify the specific features of each group of animals. And it becomes even more complex. This is a real-life picture. I took it just from my clinic 1 week before I left for Australia. This is a 87-year-old lady, nearly 19 years old, imagine. She comes to my clinic, and she has already been diagnosed with an anxiety disorder, and she's in psychiatry for about 5 years, and they're giving her lots of oxazepam because she's so anxious. And what she does, when she enters the room, she shows me her leg just like that. She didn't pose for me, it's just a spontaneous picture. And she's asking, pointing at her hematoma on the leg, is this cancer? And what she's doing during the whole day, 1,000 times, she's asking to everyone, especially her husband, is this cancer? I'm so afraid of this cancer. Could this be cancer? Am I going to die? Is this cancer? And this made me, I want to align this example with the recent paper that we wrote with the International Working Group on Right Temporal Variant of FTD, where we found that there are many, many complex symptoms in this syndrome that are potentially overlooked by both neurologists and geriatricians and psychiatrists. Because the symptoms that we found were most prevalent were a multimodal knowledge loss for verbal information. So if you have a knowledge loss for verbal information, somebody's talking about a strawberry and you don't know what a strawberry is. That's already weird. But if it's non-verbal, it's raining, and you don't know what the droplets are that are falling on your shoulder. And I think of the many patients in nursing homes who are not willing to shower. What would that be? Would that be unwillingness? Or would that maybe be non-verbal semantic loss for shower, which makes it feel unfamiliar and the patient not wanting to go under the shower? So I think we really have to be aware of those things. So this lady, she had right temporal atrophy on her scan, and I think we could explain her complaints like that, very stereotypical and not knowing anymore what a hematoma meant. There's a lot of overlap, so, with these psychiatric disorders, and I can give you a full list. There's depression, bipolar disorder, obsessive-compulsive disorder. They have many similarities, but also a few differences. For example, people with OCD are generally anxious, while people with stereotypical behavior in FTD are not anxious. But behavior variant FTD and primary psychiatric disorders, they involve the same neurological anatomical networks. That's why they have similar symptoms. So imagine if we could find a strategy, a therapy for improving, let's say, the salience network. Then we have, across disorder, magic bull. We've been trying to differentiate FTD from primary psychiatric disorder with the DIPA group, including Australia as well, together with Italy, Germany, and Canada. And we found that there are some means to differentiate the two, which is visual ratings or serum neurofilament, which is very pretentious. It doesn't need GFAP as an addition anymore. But we also try to be more specific, and there we saw that there are much less diagnostic accuracies, unfortunately. So if we take the biomarker approach for frontotemporal dementia, we could just copy what is happening in Alzheimer's disease, where CSF tau is raised because of the tangles that leak into the cerebrospinal fluid, and the A-beta is decreased in the CSF because of the plaques that remain sticking in the brain tissue. That's the theory. So why couldn't we do then just like, okay, there's a TDP aggregate, the cell will die, and we will measure increased TDP43 in the brain. Let's do the same trick. But you have to imagine that this is an immunostaining in FTLD TDP, where you find immunostaining, but it's very, very rare. You really have to look for it. Seems like it's gone already after that. In contrast to a staining of Alzheimer's disease, which is full of tau with the same magnification. So can we apply this logic to our biomarker development? Well, actually, some courageous people have recently published that extracellular vesicles can help to distinguish TDP43 and tau in FTD, which would be very, very important, making the pathologically driven stratification in frontotemporal dementia. But just like the study that I'm going to present here, although they have high accuracies, the design was made on syndromes that are already clearly enough during life. So if you include people with semantic dementia, PSP, CBD, you know already during life that they have TDP pathology or tau pathology. And that's the same for the study that we did in acetylated tau. Remarkably, this was increased in the TDP positive subjects, not in the tau subjects. So it's the counterintuitive finding here. But if you have a closer look, you see that on the right, there's the people who are having pathology-proven frontotemporal dementia. And if you count the dots, that's already very limited. And if you count the ones who do not have clinically PSP or TDP type A pathology, which is also genetic, then you might maybe end up with maximum 10 subjects that do have unknown underlying pathology during life, and the rest had all known pathology during life. So I think the design of these biomarker finding studies needs to be altered and needs to be focused on people who had behavioral frontotemporal dementia during life, and then later on came to autopsy, and then find out if the biomarker can classify them correctly. So TDP, important protein. There's a lot going on there. There are now actually conferences just devoted to TDP, and that's interesting, I think. It's involved in the transcription, regulation, and about 600 genes are under the influence of TDP. One of them is UNC13, and we've recently published, and it's also been published for ALS, that a specific polymorphism of UNC13A is associated with a stronger decline, so a less high survival rate in FTD. So there's a list of genes that are under the influence of TDP, and all of these might, again, have influence on the disease process. So it's becoming very complex, and again, much more complex than Alzheimer's disease. So what happens, there is a pathological process whereby actually TDP, normally located in the nucleus, let me see if that works, is now going to the cytoplasm. We don't know why exactly, but it's after post-translational modification. And then finally, there is no TDP left in the nucleus anymore. And then it ends up in broken parts, and the cell will easily die. Because TDP plays a role in alternate splicing, so you have the DNA, and then you have the RNA, and TDP actually hides cryptic exons, prevents cryptic exons to be included in the protein. A cryptic exon is a part of the DNA that should not be transcribed. It comes from viruses or other parts. When it comes in the protein, it's not good. That's something you should remember. And TDP prohibits this, but when TDP is not functioning well, you'll have all kinds of peptides, truncated or full parts, that include those cryptic exons. Part of them are being washed away, moved away, by a mechanism that regulates that proteins that are not good are being swiped away. It's nonsense-mediated decay. But some of them survive, and they're actually forming a kind of noise in the cell. So peptides that include cryptic exons. Well, the thing is, that is of course very specific for TDP loss, so if we could measure those, that could be a kind of new area to enter for biomarker research in FTD, and that's actually now taking place, but it's still very, very challenging. If we look at how to target FTD then, we have multiple approaches. At this moment, gene therapies are going on, especially in the progranulin carriers, because they have lack of progranulin. But targeting TDP, you could envisage that there are multiple approaches that prevent TDP forming clumps in the cytoplasm and destroying the processing, metabolic processing in the cell. So to conclude, I think it's important to see FTD as a spectrum of disorders that is more or less triangled between several conditions, which are all important for your understanding and also making progress in FTD. So in the first place, dementia, Alzheimer's disease. FTD causes dementia, but it's more heterogeneous, both clinically, pathologically, and it's less measurable than Alzheimer's disease. And the patients' and caregivers' perspective are very different, especially when you ask them how they want a therapy to be done. But still, FTD might benefit from placing it under the umbrella of dementia, because, of course, it's still very important that we have people coming to the clinics, memory clinics, and have specialized care. FTD has a lot in common with primary psychiatric disorders, and that distinction is so important, because if you diagnose a severely depressed patient with FTD, you will not give that patient a proper treatment that will miss, for example, ECT and being placed in a nursing home, which is a grandiose mistake. On the other way, there are also people that wait a very long time for the diagnosis of FTD. Conversely, their similarities might also open avenues for cross-disease treatment. And then lastly, FTD has common pathophysiological features with ALS. It's also associated clinically because people can have FTD with ALS or ALS with FTD. And they can both benefit for the developments in understanding TDP-43 pathology and also drug target development. And with this, I would like to thank you for having my presentation here, and thank you for listening.
Thank you, Yolande. That was wonderful. Those amazing insights. Does anyone have any questions? Please make your way to the microphones.
Thank you very much, Yolande. A wonderful talk, very, very clear. I'm coming more from the Alzheimer's side of things, and there's been some speculation that infections may be involved in Alzheimer's disease as well. There's been recent publications about vaccinations providing some prevention against dementia generally. It's hard to know whether it's dementia generally or Alzheimer's. Has there been any work with that, with either infection or the vaccination part in FTD?
As far as I'm aware, there has been not a focus on infections in FTD, as far as I'm aware.
Right.
Yeah.
And you know whether, like there's big studies showing that shingles vaccine was had 20% relative reduction in risk. But we don't know what sort of dementias people had generally. Has there been any work on looking at the role of vaccinations in prevention?
Well, it could be a suggestion that FTD is related to the varicella virus because of the location of the pathology. I don't know if there are, FTD is very rare, so I think those things that you would want to know from, for example, shingles vaccinations,
Yes.
very difficult to deduce from
Sure.
the data that are there. On the other hand, I'm not quite sure if the herpes would really be causative, because we also find traces that FTD is starting in the very large cells and is analogous to ALS, where we have the pyramidal cells and the von Connemara cells in FTD, And these are cells that make use of a lot of energy, and that could be that those affect the TDP metabolism in the first place. So that could be also infectious disease unrelated.
And if I could just add one more question, is there a pipeline of drugs in trial?
There are pipelines of drugs for genetic FTD. For sporadic FTD, this is much more difficult because people are already more advanced when we diagnose them. And the trials that are there are just focusing on symptoms or maybe very general neurodegenerative mechanisms. But unfortunately, we're still not able to have a patient with sporadic, so non-genetic frontotemporal dementia, and then stratify this person if there's TDP or tau in the brain, we don't know this, and then do disease-modifying treatment. That's not possible at this moment. Yeah.
Hi, fantastic talk. Thank you so much. I'm really intrigued by the different-sidedness of the semantic dementias, and it's so nice to see about that the right side that you have presented. Why do people get it on one side or another? Do we have any idea of why it develops in the right side for someone or the left side for someone else?
That's so fascinating. In the first place, it's already fascinating why it affects the first half of the half of the brain. On the other hand, PCA is really, you can flip the coin, right? But why right, why left? We are now, we have submitted a grant where we want to study in more detail genes that are involved in neurodevelopment, because yeah, it could be that neurodevelopmental factors who also are related to migration of neurons in the embryonic phase are involved in FTD. We still don't know exactly. It is fascinating.
Yeah, that was a great talk. What are your clues about clinically differentiating the behavioural or the dysexecutive variant of Alzheimer's disease from behavioural variant FTLD?
Yeah, that's a very good question. In our population, we do have patients that have clinically an FTD syndrome, but they do have AD biomarkers. That doesn't necessarily say that these patients do have the dysexecutive variant of Alzheimer's disease, because we find co-pathology. And we have even cases that do have abnormal CSF without having the AD pathology in the brain. So it sometimes can even be discordant. So you should be very careful by excluding everyone who has an AD biomarker profile as having FTD. On the other hand, there are patients with Alzheimer's disease who do not have FTD but are very similar as the phenotype. And we found in former studies that the phenotype is a little bit intermediate between typical Alzheimer's disease and behavioral variant FTD. But still, yeah, this is a matter of debate, and it's very hard to predict this clinically.
I have a question, and that was a brilliant talk. Thank you so much. This might be a silly question, but I'm struck by how TDP43 is involved in frontotemporal dementia, also in ALS or motor neuron disease, and also been recognised as a cause of dementia with amnestic presentation in people aged over 80, quite common in different locations in the brain in each syndrome. Do we have a sense of what pathophysiological process is driving this TDP43 in these different locations and at different times?
Yeah, that's also a very good question. Why does TDP spread in the hippocampus in late, and why in the frontal lobes in FTD? And yeah, you can even argue that everything that is TDP is frontotemporal dementia. Yeah, although they don't have the behavioral problems. These are still open questions, unanswered. Yeah.
I just have one other question for you, seeing as I have you on the stage and Yolande is such an expert in this space. In terms of, from a clinical perspective, when you're faced with someone that you're not quite sure, is this frontotemporal dementia or is it primary psychiatric disorders. So what are the sort of tools that you use to help sort of tease that apart, to sort of try and reduce at least that diagnostic delay?
Yeah, from clinical experience, that's also a very good question. I think it's having the sense of what a socio-emotional disturbance is, because when you read it on paper, I've got many referrals. When I read it on paper, it's FTD, because they check all the boxes. They have disinhibition, abnormal eating behavior, as reported by the caregiver. And when I enter the room and see the patient, I think, no, this is not FTD, and why? So the socio-emotional changes is something that you have to try to grasp, and they're highly differential. As a trick, I use a few things, like, for example, can you just describe yourself? What is your personality? If people can say, well, I'm very introvert, but sometimes when I have a lot of stress, I can really explode, then that's a sign that you have some interoception. You know about yourself, how you interact with other people. So that's one. I also let them describe something about their biographical history, and usually their history is different than mine, so they have to explain me a little bit. They have to give me some context to understand their story. If people do that, like, oh, you have to understand, I'm living on a farm, which is far away from town, then people are trying to take my perspective. So that's also a sign that they have good social emotional function. So those little things, and also, for example, seeing the discrepancies in the emotions with the partner, asking to describe how did you think that your partner would feel at this time. So they're just clinical tricks, but you can never be sure for 100%.
I think that's perfect timing. So, yeah, thank you for an outstanding keynote presentation. That work reminds us just how diverse sort of neurodegeneration is, and highlights the importance of how we have to think differently and think laterally, I guess. So thank you for sharing your expertise and insights. I now invite the ADRF convener, Associate Professor Stephanie Ward, for the final awards and closing. And just one final note from everyone is to thank you, Stephanie and ADNET, for such an inspiring and fun meeting.
Thank you, Emma. Thank you, Lando. That was a brilliant presentation. Okay, now, one of the highlights of ADRF every year is the opportunity to recognise the outstanding research presented by our delegates through the poster program. I think the calibre this year has just been fantastic. And it really reflects the breadth and strength of dementia research taking place across Australia, spanning from basic science to prevention, diagnosis, treatment, post-diagnostic care and community research. On behalf of ADNET, I would like to congratulate all poster presenters and thank them for sharing their work. And it is now my pleasure to announce the recipients of the ADRF2026 Best Poster Awards. Best Poster Community Research. The award for Best Poster Community Research goes to Lucy Luaka for her poster, Olives in Tomato Sauce, Joy Meets Culture. Congratulations, Lucy. The award for Best Poster in Basic Science and Discovery goes to Sophie Mattis for her poster, tau and TDP43 specific grey and white matter degeneration in frontotemporal lobar degeneration. Congratulations, Sophie. Best Poster Diagnosis and Treatment. The award for Best Poster in Diagnosis and Treatment goes to Ishara Paranawa Withana. And this award recognises his poster, Higher Clinical Throughput, Quantitative Amyloid Beta PET, using abbreviated acquisition windows, correction factors for 18F fluorobetapia, 11C Pittsburgh compound B, and 18F NAV 4694. All right, Best Poster in Prevention. This award goes to Laura Dodds for her poster, Behaviour Change Trajectories in a Rural Dementia Risk Reduction Trial. Congratulations, Laura. And our final poster award is for post-diagnostic care. And the award for Best Poster in Post-Diagnostic Care goes to Den-Ching Angel Lee. This award recognises her poster, Referrals to Allied Health Professionals for People with Dementia, an analysis of general practitioner data from 2 Australian primary health networks. Congratulations, Angel. I think maybe Angel isn't here. Is any member of her team here that perhaps might like to accept it on her behalf? Okay, well that actually draws ADRF 2026 to a close. And I wanted to share with you that this is actually the first time I've had to have full responsibility of convening a conference. So I was coasting along okay until about 3 weeks before the conference started, when suddenly panic hit. And I had this overwhelming sense of anxiety. But I'll tell you, my anxiety didn't last very long, and that's really for 3 reasons. So first of all, this conference has really been made possible by the tireless and thoughtful work of the ADNET Central Governance team, led by Jerry Santos, Alida Apostolasis, Dale Amsterg. We've had support from Matilda Busquet and Melanie Pierre-Louis. And just so you know, we've not had a professional conference organising committee involved. And these guys have worked tirelessly to make all of these conference possible. So I knew we were in safe hands, and perhaps we could all just give a round of applause to the team. I've also had the support of an amazing organising committee, and that's included Karen Anstey, Michael Braxby, Henry Brodati, Tanya Buchanan, Jürgen Goetz, Colin Masters, Ralph Martins, Sharon Naismith, Moana Sawan, and James Vickers. So thank you very much for all of your contributions. The third is again to acknowledge the work of the volunteers who have been running around with their green t-shirts, many of which are young researchers themselves, and I acknowledged yesterday and I forgot to write their names, but thank you so much. But actually the fourth reason I stopped panicking was 3 weeks ago when I started to panic. I started to review the program again. And I was really blown away by the quality of what this meeting contained. We've had 3 incredibly outstanding international speakers, Yolande today, we had Brendan Lucy and Craig Ritchie. We've had 3 outstanding local keynote speakers, Parminder Sachdev, Professor Jane Alty, and Dr. Claire O'Connor. And we've had this really incredible program of oral presentations and posters. And honestly, every session I have been to, I've learned something, I've been inspired, and I've especially been inspired by people who are early in their career presenting really exciting work. And I'm definitely leaving this meeting with a lot more hope than I think I came into this meeting. Because I think we can recognise we've still got a lot of work to do, but we can also see that we're making a lot of advances from understanding disease pathophysiology to new treatments, to implementing new treatments, and to not just medical treatments, but allied health interventions as well. So I want to thank you all for making the last 3 days what I think is the best ADRF ever. And I'd like to say that I hope to see as many of you as possible in Perth for ADRF 2027, where ADNET and the Asian Society Against Dementia will come together to welcome colleagues from across Australia, Asia, and beyond. So thank you to all of you, and hope to see you next year.